everyone, and welcome to my talk at ASMB called Deciphering Programmed DNA Elimination in Mesoroptytis nematodes. So I'm Brice, I'm a postdoc at CNRS and ENS here in Lyon, and I'm working in Marie Delatre's team. So the menu for today is I'll introduce you to programmed DNA elimination, which I'll abbreviate as PDE. First at the level of the biological discovery, and then at the level of the bioinformatics and detecting it and the data we have. I'll show you the results I have and then I'll end with some perspectives. So to start with the biology, the basic observation is that some species actually possess distinct germline somatic genomes. And this was first observed early on in late 1800s by Theodore Boveri, looking at this large parasitic nematode. And what he observed is that if you look at the cells in the gonad, so the cells from the germline that give rise to the next generation, and these are the ones in the red gonad here, you have a single large pair of haploid chromosomes. But if you look at the rest of the cells in the rest of the body, so the soma, those that don't give rise to the next generation, you've got more than two single pair of chromosomes, and they're reduced. So overall, actually, you've got a fragmented and reduced genome. And what he also observed is that this occurs during early development in these worms. Now, it was subsequently and since observed in many different animal clades, including fish, mammals, birds. So it's actually quite widespread across the tree of animals. But no real lab tractable models have been available until recently. So in fact, we don't really know anything about PDE. We don't really know what it's for, why it exists, how it occurs, so what the proteins involved are, nor how it evolved. And so what we're working on in the lab is a species of nematode as well that does undergo PDE, but we can actually culture it in the lab and manipulate it in various ways. So this is what it actually looks like, well, schematically, in a specific nematode that we study called Mesoroptytis bellari. So this is a representation of a 5-cell stage embryo, and you've got the cell that's going to give rise to the gonad and all sexual cells over here, so the germline progenitor cell, and here are all the cells that give rise to the soma. And this cell in particular, at this stage, is undergoing a division of elimination. So what this looks like, schematically again, is here I'm showing a pair of two homologous chromosomes. In truth, there's actually 2 n = 20, so 10 haploid and 20 diploid chromosomes in bellari. And they're replicated, so they're ready to go through mitosis. And what you can see is the telomeres at the extremities. These are these repeat sequences at the end of chromosomes that are necessary to maintain karyotypes. And what happens is, for all chromosomes, you've got these fragmentations at the edges of the chromosomes. And then after mitosis, you get segregation into 2 daughter cells, and this is one of the 2 daughter cells. And what you can see is that inside the nucleus, so this is the nuclear membrane here, and this is the cell membrane, you've got the reduced version of these chromosomes having been segregated into the daughter cell. And what happens is, if you look at the cytoplasm, you can see these fragments occurring. Then after a few cell cycles, these cytoplasmic fragments are degraded, and they disappear completely. And telomeres reappear on the retained DNA, because telomeres are necessary for proper chromosome maintenance. And it's not exactly just the extremities that get fragmented. You also get internal sites of elimination, which lead you to from 20 diploid chromosomes to over 40 in the somatic cells. And then this actually happens in all the somatic founder cells, so all the cells in white here, between the 4 and 8 cell stages, meaning that the whole of the body has a fragmented, reduced genome. Right, so that's for the basic biology. Now, what does it look like bioinformatically? So there's sort of 3 questions that you can answer reasonably easily if you have good data bioinformatically. And, those questions are pretty fundamental. So one is, are these locations where the breakages are happening sharp, i.e. are we cutting to the base, or are they fuzzy, i.e. are we cutting maybe at different places, slightly different places across different cells in the embryo, or across different embryos as well. Now, if we can reconstitute the actual sequences that are cut, one obvious question is there a motif associated with the break site? And then, downstream, can we actually identify the proteins that are affecting the breakage? And then, of course, if you can reconstitute the two genomes, so the germline genome and the reduced somatic genome, the question is what exactly is eliminated, right? How much of the genome, where on chromosomes, and which elements? So now I'll go into a bit of data that we have. This is not everything we have, but this is what I'll show you today. So we've got collaboration with the Sanger Institute and Tree of Life, where they've generated for Bellari here what they usually use to build these sort of chromosome level assemblies in Tree of Life. So this is both PacBio Hi-Fi and Hi-C for scaffolding. PacBio Hi-Fi, you're looking at reads of about 16,000 base pairs on average, or the median actually, and they're very high quality. So you're looking at 10 to the minus 3, 10 to the minus 4 sequencing errors per base pair, which is on par with Illumina, meaning very high quality. Coverage is not that great, it's about 30x. And what they've done actually is they've sequenced a mixed population. So they've just taken a whole set of Bellari worms. And that includes embryos, young adults, so these L1 larvae over here, and fully fledged adults. Now, one issue you actually hit when you sequence these mixed populations is if you look at a L1 larvae, for example, so a young adult, it's only got one germ cell, so it hasn't actually developed its gonad yet, versus 500 somatic cells. So essentially, all you're going to get is the reduced genome in blue over here. Yeah, and you're mostly going to miss the germline restricted, i.e. retained, and germline specific DNA in red here. Now, if you sequenced specifically adults, and actually more specifically male adults, you get a much better ratio of germline to soma. These males here are packed with sperm cells, and so that you're looking at more like 300 germ cells for 1,000 somatic cells. So you're looking at a much better ratio, about 30% of germline DNA. Fortunately, as they've sequenced a mixed population, we've only got about 3x in total of germline in this data. But we've got new data, new Hi-Fi, where they've actually enriched for adults, and they've got more germline. I'll just mention that later on. So we also have Hi-C, which is actually gives you information on DNA proximity. So any sort of adjacent molecules in 3D space will be sequenced together. So that's a good way of actually reconstituting full chromosomes. Got plenty of coverage on that. It's also on a mixed population though. And what we did in our lab is we actually handpicked a set of males and also females and sequenced those with Illumina. So we've got about 100x, for example, for a male of 250 base pair paired end reads, and about 30x of which is germline, so it's much better. Now, I've alluded to the fact that, we expect, and we see from cytology, we've got 20 diploid chromosomes, so 10 haploid. So ideally, an assembly of the germline genome should reach 10 chromosomes, okay? Because we usually assemble a single haplotype. And what we actually get from combining data 1 and 2 is a nice set of 23 assembled chromosomes. So this is a Hi-C matrix. So the redder the more red you have in a given position in this matrix, the more Hi-C contacts you have. So the more DNA contacts in 3D space you have. And you can see that you got 23. You have 23 squares here with very nice contacts in the diagonals, and not much contacts between different squares. So this clearly indicates we've reconstituted 23 physical units of DNA. And what we reckon has happened is, because we have so little germline, essentially we've assembled the soma. So we've assembled the fragmented reduced genome. And as I told you from cytology, we know that there's more than 40 chromosomes in the soma, and so this would correspond to about 46 chromosomes, diploid chromosomes. So it's nicely consistent. And I told you we actually have new Hi-Fi data, 120x and much more germline, from which I'm trying to now reconstitute the full germline genome. But let's see what we can actually do from just this somatic genome. So the key thing to actually understand is how can we really reconstitute these breakpoints, the location where you've got cutting during the embryogenesis, from the current data we have. So this is a representation of the somatic genome, and this is a representation of the germline genome, so the full genome, and the purple bit here is lost, okay, from in the transition from germline to soma. And I've told you that in the soma you've got these telomeres that reappear, okay, at the end. So if you align your sequencing reads to this somatic genome, you'll obviously get a whole bunch of reads which contain telomeres and which match up to the extremity here. But you might also get, if you have enough germline coverage, these soft clips, so this bit of read sequence that's not present in the assembly and that spans over this extremity. And these soft clips won't contain telomere, they'll contain unique sequence that actually corresponds to the purple bits here. Now, of course, you can do the opposite operation. If you have a germline genome, you can also spot breakpoints by seeing soft clips that contain telomeres right at the breakpoint, okay? And that sort of corresponds to reads that come from here. But we're going to look at this case over here because I've told you we have the somatic genome. One last thing is these telomere repeats consist of units of 6 base pair called, that spell out GGC TTA, however many times, usually hundreds. Right, so let's look at the results, and what I'll show you is an actual breakpoint from Bellari. So we're looking at one of the 23 scaffolds. This is scaffold 18 over here, and we're looking at the 3' edge of scaffold 18. Now, what you can see is I've aligned 3 sets of reads to the somatic genome. This here is the PacBio HiFi data. This is the Illumina data from a single male, from a population of males actually, and this is a Illumina data from a population of females. So what you can see is I've labeled, well, this is the genome sequence, and I've labeled these telomere arrays, okay? It goes GGC, TTA, well, I've actually labeled TTA, GGC over here, and you can see that it occurs repeatedly, which makes sense. It means we've fully assembled this somatic chromosome. And in gray, you know, you probably know, but from looking at IGV, any gray bars correspond to reads that match to the genome. And then here I'm showing the sequence that doesn't match to the genome. And what we have is, across the 3 data sets, you can see that there's this sequence that pops up right at the spot where the telomeres start, okay? So the telomere array starts here, and here you've got germline specific reads that all have the same sequence across 3 different data sets. So that's quite telling. It's actually telling us that we've got a very sharp breakpoint starting right here. And that essentially what you have is the germline genome will look like this here on the left, and then a consensus of these reads over here. And then the somatic genome will have cut exactly here and started adding the telomere sequence right from here. So that's one breakpoint. If you actually look at all 46 extremities, because we have 23 scaffolds, I find 39 breakpoints like this. So what I mean by like this is sharp to the base, and where you can clearly reconstitute a germline sequence. Another thing that was nice is we experimentally validated. We took 8 with very nice coverage, and we actually validated in the lab 7 of the 8 breakpoints. So we're very confident that we've got true breakpoints. So now if we look at the actual sequences, here's a set of 25 or so breakpoints. I've placed an N at the location where you've got cutting. So this is the exact location where cutting happens. And then the second question I was asking at the beginning was, do we have a motif? So you might see if I sort of gave you long enough that actually there is something that pops up. It doesn't really happen right where the cutting happens. But you could probably see visually that to the left and to the right, over here and over here, you've got a set of sequences that are sort of consistent across different breakpoints, okay? And sure enough, if you put this set of sequences through MEME for finding motifs, you do find a nice and clear motif that occurs, and this is the red rectangles here, centered on the breakpoint in the middle, occurring to the left and to the right of the breakpoint. The rectangles in blue and green are pretty degenerate, so I didn't really pay attention to them. Now, if you look at this, it actually looks like a palindrome. A palindrome is when you read the sequence left to right and the reverse complement, you get the same sequence. So indeed, if you reverse complement this, you get C, you don't really care about this position, T, T, A, A. You don't care about this one, and G, which is the same sequence left to right over here. In truth, the motif is not exactly a palindrome, and actually these second and seventh positions do matter. And what the motif really looks like is exactly this. So you've got the cut site over here in the middle-ish, and on the left you've got an 8 base pair, a motif which has a completely degenerate position at the second position, nucleotide at the second position, but you've got a T very highly conserved at the seventh here. And conversely, here you've got a really conserved A at the second position, and your degenerate seventh position. And so actually what's happening, oh, and actually another thing that's pretty interesting is we have a completely conserved distance between the two motifs, the two 8 base pair motifs. So you have in total about between 79 and 81 base pairs between these two motifs. You might notice that actually because you have this sequence here occurring left to right on this strand, and the same motif occurring right to left on this strand, this and this sequence are actually reverse complementary, which is interesting, but we don't really know what that means at this stage. So this is what I just told you. That's what the motif looks like, and with highly conserved distances. One thing we did, which was quite nice, is we actually tested the constraints on the motifs. We asked what happens if we knock out a motif, and what happens if we change the distance. So using CRISPR, because I told you we can actually manipulate these worms in the labs, we introduced two substitutions in one of the motifs, the right-hand one here, and we observed for that specific breakpoint that elimination was abolished. So it no longer chopped up the chromosome over here and eliminated the right-hand bit. Similarly, if we change the distance, which is pretty striking, here we obtained a CRISPR mutant with an 8 base pair insertion between the motifs. Elimination was also abolished. So pretty striking. And we're currently sort of testing that further. So the bioinformatic conclusions so far, the two first questions I've clearly answered. Are the breakpoints sharp, i.e. we've cut to the base, or fuzzy? They're very sharp to the base. This means that PD is most likely actually homogeneous across cells and across individuals, so it's very reproducible biologically. And then do we have a motif? Yes, very strong motif, and I've told you it's actually highly constrained as well. Now to the question, what exactly is eliminated? We've done some work, or actually a previous postdoc has done some very nice work where she showed that it's mostly repeats that are eliminated, but you do have about 100 genes that are also eliminated, destroyed. These are perhaps unsurprisingly actually enriched for functioning in the germline. Okay, so they're most likely expressed only in the germline, which makes sense because they're absent from the soma, right? So if they were essential from the soma, in the soma, they probably wouldn't be destroyed. And as I said, I'm currently trying to assemble a full germline genome using this new HiFi data with more germline signal. So that's for another talk. So perspectives. What we're trying to do now is, still in Bellari, we're trying to identify the actual nuclease, so the protein is doing the chopping up. We've got some assays for doing this, and we're also doing more CRISPR to actually further delineate the motif sequence and distance constraints. So that's for the wet lab. I'm doing more of the bioinformatics, of course, and I'm going to try and find out what the nuclease is on my side using various known transposases and endonucleases and try and annotate them on the Bellari genome. Another bit of information we have that's very useful is we've got RNA-seq for embryos before, during, and after elimination. So we can look for genes whose expression actually peaks during the elimination stages and try and cross that information, intersect that information with any nucleases that we find. And finally, we think there may be a role for epigenetics here. So we have generated data for histone marks, chromatin accessibility, and also small RNAs, which are very important in nematodes. And we'll see if there's a sort of a role for epigenetic specification and how that may work. Now the last thing is we've got a whole comparative genomics aspect as well. So we're generating data from 4 or 5 other Mesoroptytus species. We've got some nice data, encouraging data for a sister species, Spiculigera here, where preliminary data, i.e. 2 breakpoints, shows that we've got the same motif pair actually and at the same distances. So it looks like it's conserved in this genus. But then if you further look out, and we'll do that too, in other genera from the same family, in fact, other labs are doing this and have found that different motifs occur. This time centered at the cut site as well, so it's not 2 motifs occurring either side, it's motifs occurring right where the cut site's happening. So we've got, overall we think we've got actually a nice system ourselves and other labs working on these other genera for these nematodes to try and answer this 150-year-old puzzle. So that's where we're at the moment. With this, I want to thank the Delattre Lab, which is mostly experimentalist, but I'm glad to be bringing in the bioinformatics work. Our collaborators at the Sanger Institute, very great bioinformaticians, and this is a nice picture of nematodes that spontaneously made a smiley. And thank you to our funders and you for your attention.